All right, so we have talked a little bit about mitosis and the major steps that a cell has to go through to divide in that system. So now we're going to switch gears and talk about meiosis, or the sexual reproductive cell process. And so we can't talk about sexual reproduction without also addressing genetic variation. So fossils actually indicate that about between 1.5 to 2 billion years ago, cells actually became able to reproduce genetically. And that pace of evolution really has depended upon the variation that is present in the gene pool. So, the ability to adapt to new environmental stressors. So, sexual reproduction is really made up of two major processes. The first is meiosis, and the second is fertilization. Meiosis is where we're taking one copy of a gamete and turning it into, or replicating it into, a 1N gamete. Fertilization is where we take two 1N gametes, or two 1N cells, and those two come together and create a diploid. So meiosis is broken up into two major steps, meiosis 1 and meiosis 2. In meiosis 1, we begin where we have chromosomal material, or chromosomes, within the nuclear membrane, and those chromosomes are going to have two copies associated with them. So here we've got chromosome, we'll just treat the red as chromosome 1 and the blue as chromosome 2. So we have two copies of chromosome 1 and we have two copies of chromosome 2. The result of meiosis 1 is where we reduce these 2N structures to a 1N structure. So we're only left with one copy of chromosome 1 and one copy with chromosome 2, just in two separate cells. All right, and then meiosis 2 is where we further reduce through mitosis this 1N structure to 4 1N structures. So we're going to reduce down by the simple steps that we already covered in mitosis. And the result is 4 1N structures. All right, so this slide tends to get a little bit busy, so we're just going to work through it one piece at a time. In meiosis 1, a lot of these principles are going to be similar to what we saw in mitosis. So in meiosis 1, chromosomes are going to begin to condense and the spindle fibers are going to form. So very similar to what we saw in prophase of mitosis. That's going to lead us into late prophase 1, where we have a nuclear membrane that is still intact, and those spindle fibers are actually going to start to relocate to opposite poles, very similar to mitosis. Those in late prophase 1, we're also going to see condensation of the DNA material into homologous chromosomes. So the pairs of chromosomes are going to begin to come together in late prophase 1. Also, we're going to see the chimastic actually taking place, to where we have a copy of chromosome 1 and chromosome 2 both present. They just may pair up differently. And it's at this point in late prophase 1 that crossing over takes place. And we're going to discuss crossing over in a little more depth, but crossing over is one source of genetic variation. Also, the nuclear membrane is going to begin to break down even more here. So we can see it's fairly intact in late prophase 1. However, as we progress through late prophase 1, we're going to start seeing more disintegration of that nuclear membrane. Next, our cell is going to proceed into metaphase, and this is where those homologous pairs of chromosomes are going to line up randomly along the metaphyseal plate. In anaphase 1, we're going to see those homologous chromosomes actually separate and move towards opposite poles. So here is where you can really see the crossing over process more illustrated. So because we have different ends of chromosomes associated with the opposite chromosome here, this is a representation of the principle of crossing over, to where we can have DNA material from two separate chromosomes actually crossing over and reassociating itself with a new chromosome. And finally, that leads us to telophase 1, where chromosomes are going to arrive at the spindle poles, and the cytoplasm is going to divide. That DNA is going to relax again into a big web. And we have two new cells that have two copies, or that have one copy of each piece of DNA. All right, so that leads us into the second major step of meiosis, which is meiosis 2. And we begin meiosis 2 with prophase, where the chromosomes are going to recondense. After prophase 2, we then begin, the cell proceeds to metaphase 2. And this is where cells are going to line up along the metaphyseal plate again. And then those cells are going to proceed into anaphase 2, where instead of whole chromosomes separating and moving towards opposite poles like in anaphase 1, sister chromatids are going to separate and move towards opposite poles. And finally, in telophase 2, the chromosomes are going to arrive at the spindle fibers, and cytokinesis is going to take place. That cytoplasm is going to divide and form, and what we end up with is 4 new 1N cells. So again, in meiosis, we're just keep in mind that one chromosome, or in this example that we're going to work through, the nuclear envelope is holding chromosomal material. And on these two chromosomes here, this chromosome has a big A and a big B gene. However, this chromosome, or its homologue, possesses a little a and a little b gene. Right, so as we go through DNA synthesis, or the S phase of the cell cycle, we're going to replicate that DNA. So now we have 2 copies of the big A gene, 2 copies of the big B gene, 2 copies of little a, and 2 copies of little b. So DNA replication is going to produce identical sister chromatids. All right, so then during the process of meiosis 1, we actually have the concept of crossing over actually taking place. And when crossing over takes place in prophase 1, segments of the non-sister chromatids are exchanged with one another. So normally we would have big A, but if crossing over takes place, then the big A gene and the little a gene may have switched chromosomes. This is crossing over. And then as that cell proceeds through the rest of meiosis 1 and meiosis 2, our ending result is 4 1N cells that have this new portion of DNA associated with it. So this is really one source of genetic variation that allows us as an organism to evolve. So again, as we look at meiosis, one more time, because repetition is key, we're going to start with middle prophase 1, where we have centrosomes. They're going to begin moving towards the spindle fibers. The DNA within the nuclear envelope is very relaxed. In late prophase 1, we're going to see condensation of that DNA and spindle fibers moving towards their poles. We're also going to see pairs of homologues coming together in prophase 1. Late prophase 1 is where the nuclear membrane is going to keep disintegrating, and where crossing over can take place as we exchange genetic information between the chromosomes. In metaphase 1, those chromosome pairs are going to line up along the metaphyseal plate, and the pairs of chromosomes are going to separate towards opposite poles in anaphase 2, and we get splitting of the 2 cells in telophase 1. At that point, that DNA that is contained inside the nuclear envelope, or the new nuclear envelope, is going to relax, and that relaxed cell is then going to proceed into meiosis 2, where in metaphase 1, prophase 1, we're going to see those cells recondense, And then in metaphase 2, those chromatids are going to line up along the metaphyseal plate. And finally, in anaphase 2, we see the sister chromatids, so the single copy of genetic information, being pulled towards the spindle fibers. So telophase 1 is going to result in 4 1N genotype, or 4 1N cells. So to go through meiosis again, if you need to spend a little bit more time on this because it's been a while since you've been in a 101 class, this table is a really good one to start with because it's going to give you the stage of meiosis and then line up the major events in each one of those stages. So spend some time studying this table as you work through this information for the first exam.